This is a production of Cornell University.
Yeah, it's really fun to be back here. Not only was I an undergrad here, I was a plant science major. And so I took the majority of my undergrad classes in this building. And when I saw Plant Science 404, I was like, I'm sure I've been in that room. And I heard many, many seminars in this room. So it's really fun to be here. Okay. So for most of my career, as you can imagine, being a plant science major in this department, I've been really interested in host microbe interactions. And across my career, I've been really interested in sort of the continuum and the spectrum of host microbe and host pathogen interactions. And so symbiosis, I define in sort of its classical definition, which means living together. And I'm getting a little cut off at the bottom there, but it includes pathogens, mutualists, and commensals. And so I'm going to use it to mean sort of all living together interactions, including your mutualistic symbioses. But I've been really interested in how hosts and microbes navigate this continuum. So do hosts get to decide, do microbes decide, and how, when they come together, does that interaction determine the outcome of this interaction? And so from my time in my PhD work, I learned that these changes can actually occur over very short evolutionary distances. And so Agrobacterium, our plant pathogen, and Syringomycin, a mutualist, are actually very closely related. So in the box cut off down here is Syringomycin, which is very closely related to Agrobacterium, which is also in the box. That's a plant pathogen. And if you make a synteny plot of these, I don't know if I can actually pull that down a little bit. No, that's not the way that works.
On the left.
Oh, okay, you can see it over there. Thank you.
Let's close down the bar.
Oh, okay.
I think you point at this.
I think maybe you almost got it.
Oh,
Yeah.
on this one, there we go. Too many computers and screens. Okay, good. Thank you. So if you plot the genomes of Agrobacterium here and Syringomycin, you can see that they're actually very highly conserved. So each dot is a protein coding gene. And you can see that if you line them up, they actually align quite well with a few, exceptions. But over an evolutionary distance where you can line up genomes, we have a mutualist and a pathogen. So that suggests that relatively small genomic changes can drive these major transitions in lifestyles between bacteria. Now I'm not advancing. Okay. So my interest in these sorts of questions started here in this department and often in this very building. I actually have used this slide before. I didn't even make it just for this talk. And I started working on this very famous plant pathogen, late blight, which is a devastating, pathogen, and is about as far from Rhizobia legum and AM fungi as you can get. Just a brief interlude. One of my first classes that I took in this department was as a freshman. My faculty advisor suggested that I take plant pathology. And I went into Bill's class, and the first day everybody was talking about like R genes and Agrobacterium transformation, and I had no idea what was going on. And I went up to Bill, I don't know if you remember this story. I went up to Bill and I was like, I don't think I should be in this class because I have no idea what's going on. And he was like, okay, what's your name? And I said, Cara Haney. And he was like, oh, you're Irish. You're only here because of a plant pathogen. You should absolutely be in this class. And so it turns out that that's true. And I had no idea at the time, but my own personal family history was shaped by plant microbe interactions. So my dad's family fled Ireland during the Irish potato famine in the late 1880s and immigrated to North America at that time. And so I worked in Bill's lab and so on. But from this, there's been a few things that really have stuck with me. The first is that plant microbes have shaped human history. But second, within this pathogen, a single pathogen, so I've talked about spectrum of pathogens having multiple lifestyles. A single pathogen can have multiple lifestyles. So Phytophthora is a case where they have this biotrophic stage where they look like they're not even doing anything to the plants, and then they suddenly kill the plants. Something else that I often mention from this is that You can also have tissue specificity, and I'll come back to that at the end, but with tomatoes and potatoes and late blight, sometimes you see the fruits are like perfectly healthy on the tomatoes and the leaves are dead, or the potato tubers will be completely rotten and the leaves look healthy. So there seems to be, something around tissue specificity that I have sort of kept with me through my career. So about the time I graduated, I heard a seminar by a new professor that was being recruited into the department, Maria Harrison, and I suddenly was like, there's this whole other world of plant pathogens or plant microbe interactions. And so sort of inspired by working on sort of the other extreme, I went and worked with Rhizobia legume symbiosis. And an amazing thing about this system is that you can do genetics in a way that is really difficult with potatoes, more feasible with tomatoes. So in this mutualistic interaction, you can screen for mutants that lose the ability to form nodules and that have lost the nitrogen fixing symbiosis. But here you still have rhizobia in these nodules. And so there's a lot of questions that sort of arose about how does the plant control this? How does the plant avoid rhizobia becoming a pathogen, taking more nutrients than it can afford to share? And then how do plants actually avoid being parasitized by these microbes? And then, as I mentioned, are pathogens and mutualists really so different, as these are very closely related to pathogens? And so that in Sharon's lab, I really started thinking about this continuum. And I think it's best exemplified with these alpha proteobacteria, as I mentioned, where we have Cynorhizobium and Agrobacterium as close relatives. Even more amazingly, these are more closely related to each other than they are to Bradyrhizobium, which is another nitrogen-fixing mutualist. And Cynorhizobium is more closely related to Brucella, which is a pathogen of animals. And so it turns out that plants, pathogens and mutualists have shared mechanisms to infect very different hosts. So Cynorhizobium is here infecting a bean cell. Brucella is here infecting a macrophage. And for virulence, they both require some shared conserved ancestral mechanisms that are likely there to evade or suppress host immunity. And so confronted with the same challenge of how do you protect yourself from microbes, whether it's a pathogen or a mutualist, plants and animals have actually convergently evolved similar solutions. And so here are plants. They have things like flagellin receptors that can activate defense signaling. Animals have actually convergently evolved similar pathways. And so confronted with the same challenge and given the same molecular toolbox, plants and animals can both sense pathogens and mutualists, pathogens are non-self and mount an immune response. And so microbes likely have evolved to circumvent these pathways using similar mechanisms. And so my lab thinks about sort of two symbiotic continuums. One is over actually probably evolutionary time from free living to symbiotic. So microbes, as they from the environments have to colonize hosts, they have to actually circumvent these very specialized adaptations in immunity to colonize a host. And so those adaptations might generally allow things like Brucella or Agrobacterium to be host associated. And then the specific adaptations might be more recently evolved that allow a common ancestor of Brucella, Agrobacterium, and Cynorhizobium, all host associated. And then more recent adaptations determine whether it's a pathogen or a mutualist along this continuum. And so this is a model that my lab thinks about a lot. First, how do you become symbiotic? And then once you're symbiotic, how do you become a pathogen, a mutualist, or a commensal? And so today I'm going to tell you 3 stories from the lab. One thing I'm not going to talk about is sort of common innovations that allow symbiotic ancestors to become symbiotic, although that's something we're really interested in. I'm going to talk about once you're symbiotic, how bacteria can move along this symbiotic continuum of lifestyles from pathogenic to mutualist. And so what are the mechanisms that drive those transitions? Second, does the host have any say? So can hosts actually detect and enrich for specific beneficial microbes? And can they do so without enriching for pathogens? And a similar question, are hosts able to distinguish closely related pathogens and mutualists in the context of the microbiome, where things like effectors might not be playing as much of a role? So as Maria suggested, my lab uses Arabidopsis as a model for microbiome studies. And for many of the same reasons that you all use it for immunity studies, or those of you that do use it for other host pathogen interactions. It's a great system for forward and reverse genetics. It's our validated reference plant, so much of what we've learned in immunity or flowering time have translated to other crop plants. Importantly, the microbiome community of Arabidopsis at a very high taxonomic level resembles crops. And so it is a non-nodulating, non-arbuscular plant. But if you look at the microbiome composition, at least at the phylum level, it's very similar to our crop plants. So this is rice and corn here. And then I always like to remember that Arabidopsis is a wild plant. It's not domesticated. And so the natural variation and the tools that we have in Arabidopsis are Arabidopsis adapting with its local microbiota to its local environment. And we haven't necessarily lost those interactions during domestication as we potentially have with some of our crop plants. And so when I started working on the microbiome and trying to ask questions about sort of microbes moving along this continuum, I sort of took a page from all that I learned in this department and the power of things like Pseudomonas syringi Arabidopsis defines, or Pseudomonas syringi spedo, to find effectors and plant disease resistance genes. And I decided to focus just on a single microbe associating with Arabidopsis, and we use Pseudomonas fluorescens. And the reason is that it's enriched in the plant rhizosphere, so it's really good at colonizing plants. Is beneficial to plants, so it can protect them from pathogens. And so we know the outcome of that interaction matters. When it's there, plants are more disease resistant, and they also promote growth through a variety of nutrient and hormone related mechanisms. It's really interesting because its function can't be inferred from taxonomy. So if you look at a tree of Pseudomonas and then try to predict what they're going to be doing, it actually doesn't work. And so that makes it really interesting for these sorts of studies, and it's genetically tractable. And then Pseudomonas, like the alpha proteobacteria, actually shows those patterns of host association and lifestyle transitions within a single genus. And so within Pseudomonas, we have Aeruginosa, which are opportunistic pathogens of actually plants and animals. There's reports of Pseudomonas aeruginosa causing disease under very stressful field conditions like heat waves or poor soil quality. We have Pseudomonas syringi, which you all know a lot about, I'm sure, that are plant pathogens. And then Pseudomonas fluorescens, which are described as both plant and animal symbionts and free living. And so within this group, we study them because first they're all host associated. And Aeruginosa is really fun because it's both plant and animal associated. I'm not going to talk about that work today, but we consider that a lot. Today, I'm going to tell you about Pseudomonas fluorescens. And so we wanted to understand across this continuum from mutualist to commensal, so something that's not like living with plants, not necessarily causing harm or benefit, how quickly can those transitions occur within this group? And so we started, this is actually the first postdoc to join the lab, Brian Melnick. He asked within this group of actually well-described mutualistic Pseudomonas, so these are plant growth promoters, do they all promote growth, or do some of them not promote growth? And can we sort of understand the genomic changes that happen over these short distances that would drive that? And so we started with our favorite lab model strain is WCS365. It's a nice plant growth promoter. Within this group, I just want to point out for anybody that does 16S sequencing, these are 99% identical by 16S. So these are very, very closely related. And often some of these would be called the same ASC. So basically indistinguishable by 16S. And then Pseudomonas sort of species complex would be this whole group, Pseudomonas fluorescens species complex. And so what we found that was quite surprising was not only did not all of these strains promote growth, some of them kill plants. And so Ryan, actually one of the first strains he tested in the lab was this N2C3 strain, which is within that brassicacearum group. And not only did it not promote growth, it kills Arabidopsis under germ-free conditions. I spent a long time being convinced that Ryan just didn't know how to grow plants and it was something he was doing. But when he did this with controls of other strains that do not kill, it's clearly something unique about this strain. I'll show you a few graphs like this that are just death quantified. So here's our N2C3 pathogen. And then these, under these conditions, actually don't promote growth super well because this is rich media, but they do cause changes in lateral root architecture. It turns out that N2C3 is a quite broad host range pathogen. So we tested it on, this is rice. We tested it on tomatoes and poppies and whatever people had lying around in the lab to put in their gardens, and it basically killed everything that we could find. And so Ryan actually came from a genomics background. And as part of what he was doing in the lab, he was building a comparative genomics pipeline to try to identify genomic traits that would be unique to one strain and not another. And so the idea here is that if we take the genome of our beneficial strains and compare to the genomes of our pathogen, we should have most of it the same, our core Pseudomonas genome, but the genes required for pathogenesis or mutualism or commensalism should be unique to each of those strains. And so he built actually a Pseudomonas pan-genome where he did all by all comparisons and then just generated one giant Pseudomonas genome that now we can compare each genome to. And that makes this a much easier problem. And these tools are available if anybody is interested in the Pseudomonas pan-genome. But when he did this, we found a very conspicuous 143 kb genomic island that was unique to our Pseudomonas pathogen. That was inserted in otherwise a very conserved region of the genome. It turns out it encodes syringomycin, syringopeptin, which you may have heard of because it was first described in Pseudomonas syringae. And so these are toxins that cause disease on many plant hosts. And so this is Pseudomonas syringae of bean that causes disease in a syringomycin, syringopeptin dependent manner. So these are mutants. This is the wild type strain, mutants that don't make the toxins, disease goes away. Here's another Pseudomonas syringae on cherry. Again, this syringomycin, syringopeptin mutants no longer have a reduction in disease. And so we deleted these in our pathogenic strain, and you can see syringomycin and syringopeptin work synergistically to cause disease on Arabidopsis. So if you delete either of them, you lose disease. And so that seems to be responsible for disease. But this was kind of surprising. This is a Pseudomonas fluorescens, not a Pseudomonas syringae. And so we went back to our Pseudomonas tree, and it turns out that within Pseudomonas fluorescens, these genes actually occur quite frequently. And so here's Pseudomonas syringae. And then these are all kind of the Pseudomonas fluorescens clade at large. And you can see that there's some like Pseudomonas fuscovaginae, which is known to cause rice sheath rot, that fall within the Pseudomonas fluorescens sort of species complex. And then some are actually very closely related to our pathogen that we found, like Pseudomonas corrugata, causes tomato pith necrosis, are squarely nestled in this fluorescens clade. And so it seems that this is actually quite widespread within Pseudomonas and is moving around. So we wanted to understand more about the inheritance in this group because it looks kind of interesting. We have one group that looks like it's got it, one group that hasn't. Was this kind of one horizontal transfer acquisition event that was just kind of lost in a few strains here, or is something more complicated going on? And so we took advantage of the vast genomic information available for Pseudomonas. And if you zoom in just on that brassicacearum clade, so these are all 99% identical by 16S. And then this is the presence of that syringomycin, syringopeptin island. You can see that there's been multiple acquisition and loss events over a very short evolutionary distance. And so there's not like, there was probably an acquisition event, here that resulted in all of these strains having it, but there, you can't explain this pattern here without multiple gain and loss events. And so it seems like this island is moving being gained and lost very readily throughout Pseudomonas fluorescens genomes. And just to sort of validate that this gene or gene cluster indeed predicts virulence, we got our hands on a number of strains that have or don't have the island. All the ones in green don't have the island, the ones in red do. And it seems to very closely correlate with virulence. And so we're really intrigued by this, this idea that maybe you could have a lifestyle transition over, not the distance of Agrobacterium to Cynorhizobium, but just over, a single strain gaining and losing a single island. And so we wondered, is that just it? Is it an island that's moving around and causing these lifestyle transitions? Or is it something more complicated? And there's a beautiful example from Jeff Cheng's lab showing that Rhodococcus, through gaining a single virulence plasmid, can actually transition from a plant commensal, so associated with commensals, to a pathogen, just with acquiring a single plasmid. And so we wondered, are there other genomic changes that correlate with lifestyle, or is it just the presence absence of this island? And so we did a genome-wide association where we looked for genes that correlated with presence absence of this island. And to our surprise, we found several genome-wide changes. And so this is our syringomycin syringopeptin island here in red. And we found two additional, they were annotated as putative pathogenicity islands. We still don't know what they're doing, but at least one might be a toxin that are nearly perfectly correlated with our lipopeptide. And then we found two islands and a putative effector that are nearly perfectly inversely correlated. And this is probably too small to see, but this one here is actually a type 3 secretion island that is almost uniquely present in the commensal, not the pathogen. And then this is DAPG, which is a known antifungal compound made by Pseudomonas that's been implicated in suppressive soils. And so we have, two sort of configurations of the genome, one with this syringomycin syringopeptin, and then one with type 3 secretion and DAPG. And so we think that Pseudomonas has kind of converged on two solutions for plants and actually fungal association. So I didn't mention, but the syringomycin syringopeptin lipopeptide islands is also antifungal. And so one version is lipopeptides plus these two pathogenicity islands, and then the other is type 3 secretion plus DAPG. And all strains that have these, they end up in the same genomic location, and we have evidence that they're being gained and lost through homologous recombination. And so it seems that Pseudomonas fluorescens has two lifestyles that are transitioning over very short evolutionary distances. And so it seems that even within very closely related plants, associated strains, you can get these transitions with just gain and loss of a single island. So now I'm going to switch gears a little bit and talk about the sort of role of the host in this. And so do the hosts have any say? Can they specifically enrich for beneficial microbes or even specific groups in microbes? And then as they're enriching, how do they filter out which ones are good and bad? So plants do something really cool, which is that under certain stress conditions, I think my slides got missing a slide. Okay. Maybe I got shuffled here. So plants and animals, it's known that a normal microbiome structure is really important for host health. And so our gut, if we end up with an altered microbiome community that can make us really sick, we're associated with more susceptibility to autoimmune diseases and pathogens. And the same is true for plants. So the slide I'm missing, you would see that if there's a plant that's missing, actually it's a big component of its innate immune system, you can get disease from microbiota. Plants also do something cool where they can specifically enrich for beneficial microbes. And so the classic example of this is suppressive soils. And something I learned about in plant pathology class, there were two mechanisms of disease resistance, our genes and suppressive soils. Suppressive soils were not well understood, but this is a phenomenon where upon sensing a pathogen, plants can enrich for groups of microbes that can suppress that pathogen. This hasn't been described in animals, and it turns out it's not unique to biotic stress. Plants can also do this in response to abiotic stress. And so there's been some beautiful work from Devin Coleman-Durr and the Sundaresan lab at UC Davis, showing that under drought stress, plants also shift their microbiome. And so this is drought-stressed grass plants. You get this massive enrichment of streptomyces, and like bacteria in suppressive soils, streptomyces are sufficient to protect against drought stress. And so this, both suppressive soils and the drought example suggests that under biotic or abiotic stress, plants might actually be able to dynamically enrich for these beneficial microbes to help protect against those stresses. And so we're really interested, can plants enrich for Pseudomonas under conditions where Pseudomonas would be beneficial? And if so, can they tell apart? Can they enrich for just our beneficial Pseudomonas and not the pathogenic ones? And so to get at this, genetic screening with microbiomes has been very difficult because microbiome phenotypes are often hard and labor intensive to screen for. And so what we decided to do was to take advantage of a mutant collection that had been developed in Fred Ausubel's lab. And this is a really unique mutant collection because it was screened for root immunity phenotypes. And so these are mutants that when you add a plant hormone that normally suppresses root immunity, so pattern-triggered immunity, they're actually insensitive to that hormone response. So the details aren't super important for these purposes. The original paper was described here. But the point is that these mutants are specific to root immunity mutants involved in pattern-triggered immunity. And so we had a collection of uncharacterized mutants, and we rescreened them for ability to support Pseudomonas fluorescens in monoassociation. And we do this by growing Arabidopsis in 48 well plates, and then we can add Pseudomonas fluorescens expressing GFP, and then read Pseudomonas fluorescens GFP with a plate reader as a proxy for Pseudomonas abundance. And so this is a really easy way to quickly quantify Pseudomonas. And so doing this, we found that our mutants, we found 3 that have higher levels of Pseudomonas, and we found 3 that have lower levels of Pseudomonas. And so we have preprints about these 2, and I'll tell you about HSM13 today that has more Pseudomonas. Okay, so this plant mutant HSM13 has more Pseudomonas in monoassociation with just Arabidopsis-Pseudomonas. And so the first thing that we did was to take it and plant it in natural soil and see if it also had more Pseudomonas when there's a complex community present. And it does. So this is Pseudomonas fluorescens gets its name because it's fluorescent under iron limiting conditions. And so HSM13 has about 10 times more Pseudomonas fluorescens per gram of root than wild type plants. And this is bulk soil, which has relatively low Pseudomonas. And this is quantified here. So this is CFU fluorescent colonies per gram of root. We can also do microbiome sequencing to look at the entire community and see if is it just Pseudomonas that's different or are there larger community changes. And here we actually found that wild type and the HSM13 mutants look quite similar. And so you can see this is a principal component analysis and they separate along this axis, although that's a smaller portion of the variation than this axis, which explains bulk soil versus the plant genotypes. But when we look at the taxonomic levels, they're actually quite similar. So this is phylum level. And then when we looked at individual taxa that were different and explained the difference we see here, it was largely limited to this family Pseudomonidae. And so this is a relative abundance plot. There are some other taxa that are different, but the most dramatic change was actually enrichment of Pseudomonas, where we have way higher in HSM13. Interestingly, there's some signatures of dysbiosis in this mutant, although it's very Pseudomonas specific here. And there's much less diversity. So we get a drop in total bacterial diversity by number of OTUs and also Shannon diversity index. And so we wondered, is this microbiome from HSM13, is this a beneficial community or is this sort of a dysbiotic community that could make plants sick? First author of this paper, Yi, did a really clever microbiome transplant experiment where he grew either HSM13 or wild type plants in natural soil. And so he let the plant shift the community, so enrich for whatever it's going to enrich for. Then he removed the plants and put wild type plants in both soils, and then asked what the effect of just the soil is on plant health. And in doing this, he found that the wild type plants in wild type soil were much smaller than wild type plants in the HSM13 soil. And you can see this correlates quite nicely with increased Pseudomonas fluorescens. So there's more Pseudomonas in these soils, and so you get growth promotion. And so it seems at least that the HSM13 microbiome isn't harmful. under these conditions, although we're enriching for Pseudomonas, and I'll come back to that later, because here I'm talking about Pseudomonas as one group of things. So we were able to map HSM13, and it encodes FERONIA Kinase. So HSM13 has a point mutation in the kinase domain of a actually well-studied kinase, receptor-like kinase called FERONIA. As you can see, HSM13 is FER-8, which tells you that 7 other people have found FERONIA out of screens. And so it's a very well-studied RLK. And I won't go into the details because it's published, but this is just showing that it indeed is FERONIA. So what does FERONIA do? It is a master regulator of immunity and development, which is great in not great in many ways, because then you have to kind of filter through all of the ways that FERONIA could be affecting your particular phenotype. And so it does have a role in development. We were able to kind of uncouple that from our role in microbiome. So I'm not going to go into that in detail, but in immunity, FERONIA has a few known roles. So one is that it's known to interact with BAC1 and FLS2, which are receptors for microbe-associated molecular patterns. And so we thought maybe if you lose pattern-triggered immunity, you would get increased Pseudomonas. Second, it's known to be a negative regulator of MYC2, which regulates jasmonic acid signaling, which antagonizes salicylic acid. And so that could also result in increased Pseudomonas fluorescens. And then it also regulates basal levels of ROS in roots. So this is reactive oxygen that's just involved in normal growth and development. It's not your ROS burst inducible ROS. It's just ROS that's signaling and involved in like cross-linking cell walls. And so we thought that that reactive oxygen regulation could also be normally inhibiting Pseudomonas. And so we tested mutants in all of these pathways to try to see if loss of any of these pathways affected rhizosphere Pseudomonas. And actually, we found that knocking out pattern-triggered immunity in single mutants does not affect rhizosphere Pseudomonas. We have now tested a BAC1, SERC1, BBC, BAC1, BIC1, SERC1 triple mutant, and you get a very slight increase, but it still doesn't explain the FERONIA phenotype. Jasmonic acid signaling has no effect. It was actually only when we tested RBOH, D, and F, which are involved in reactive oxygen production, that we saw an increase in Pseudomonas that's starting to reach the level of the FERONIA mutants. And then this is reactive oxygen staining. So this is wild type, and then this is the FERONIA mutants. And then these are the wild type and FERONIA mutant with Pseudomonas. And so again, you can see that the level of reactive oxygen very nicely correlates with the level of Pseudomonas in the roots. So the green here is more reactive oxygen. And then when we have less reactive oxygen on FERONIA, that's when we have more Pseudomonas. And so to test if this was really the pathway that was involved, Yi overexpressed this RAP2, which is downstream of FERONIA, to ask if this could bypass the need for FERONIA and regulating reactive oxygen. And so here's our parental line, and then the HSM13 mutants. And if you overexpress this RAP in the HSM13 mutant, you have a dramatic reduction in the amount of Pseudomonas, suggesting if you increase the level of reactive oxygen, you can actually decrease the level of Pseudomonas. And so that suggests that indeed it's this pathway and not something related to PTI that's regulating rhizosphere Pseudomonas. So interestingly, it turns out that fungal pathogens and nematodes can make the ligand for FERONIA. And so there's 2 beautiful papers that show that nematodes and fungal pathogens can make RALF-peptides, which are mimics of RALF-peptides, which are the ligand for FERONIA. And so they likely do this because by messing with FERONIA, they can actually mess with the JA pathway and promote susceptibility to themselves. So jasmonic acid is required for resistance to both fungal pathogens and nematodes. And so we thought that was really cool because this could potentially be a way for plants to sense these fungal pathogens and nematodes and then enrich for Pseudomonas, which has been implicated in suppressive soils. And so we asked if just adding RALF-peptides, so the ligand for FERONIA, was sufficient to recruit Pseudomonas. And we did this in a couple ways. So first, we tried adding exogenous RALF-peptides. As you can see, there's at least 33 of them, so there's a lot. We just picked a couple that had been implicated in immune signaling and were like the fungal pathogens. But we've got a small but significant increase in Pseudomonas with one of them, RALF-33 here. And this was dependent on FERONIA. So you don't see the increase in the FERONIA mutant. And we also overexpressed one of the RALF-peptides, and you get an increase in Rhizosphere-Pseudomonas just with the presence of the ligand. And so we think that fungal and nematode pathogens may make RALF-peptides, and they do so with the goal of inhibiting jasmonic acid signaling and promoting susceptibility to themselves. And then plants may be guarding this pathway by downregulating reactive oxygen, basal reactive oxygen levels, which in turn results in more Pseudomonas that could protect them from these fungal pathogens. Okay, so I think I've got 10 minutes, is that right? Okay. Okay, so I've told you two stories. One of the, that bacteria can transition over short evolutionary distances, and then that plants can sort of generally enrich for Pseudomonas that results in benefit. These two things are kind of, even in the lab, a little bit at odds with each other, right? So I have Pseudomonas as one group that's all beneficial, and then Pseudomonas where we have pathogens and mutualists that are super closely related. And so a question that I think naturally follows is, can plants tell these apart? When they're enriching for Pseudomonas, are they able to just enrich for the beneficial ones? And so we first asked this using our FERONIA mutant to ask, in this mutant, are you only enriching for beneficial Pseudomonas? And so here we have Pseudomonas commensals, Pseudomonas pathogens, and then non-Pseudomonas. And then in green are wild type plants, and blue are FERONIA mutant. And so you can see commensals all enriched in FERONIA, with the exception of this one, which I think is super interesting and we don't know why. But the pathogens are also enriched in FERONIA. So this is Pseudomonas syringi here. Some of our opportunistic pathogens like the N2C3 I told you about, and Pseudomonas aeruginosa. And so it seems like it can't tell them apart, at least in this mono association, that FERONIA is enriching for pathogens and mutualists. However, Pseudomonas are not great soil pathogens. And so if anybody knows of cases in the literature where Pseudomonas are root or soil pathogens, I'd be really interested. But it seems that generally they're leaf and stem pathogens. And so just to sort of illustrate this, on plates, if you add DC3000 to Arabidopsis roots, right, none on the leaves, you get stunting of plants, even with DC3000. And then this is our opportunistic pathogen. But if you take Arabidopsis and you grow them in natural soil and you drench the soil with DC3000, you get no disease. And we have tried, and every time we see a spot on the leaf, it's probably that it just kind of splashed onto the leaves. And so that suggests that maybe there's something about the soil community that is protective. And so this is a master's project from a student in the lab who asked if just in binary interaction, so if you put a commensal and a pathogenic Pseudomonas on the same plants, do you get disease? Or can you protect from the pathogen? And so this is N2C3, our pathogen. And Nicole found that many commensals and many that are closely related to the pathogen. So here's the pathogen on our tree. And then this closely related commensal, if you mix them on plants, actually the commensal wins. And so they protect against the pathogens. This isn't true for all commensals. So this is another clade of Pseudomonas fluorescens here. And if you mix those, they actually don't protect against the pathogen. And so this suggests that either some of the commensals can outcompete the pathogen or kill the pathogen, or the plants actually can somehow enrich for just the commensals and not the pathogen. And so we first tried to see if there was direct antagonism. So can the protective commensals directly kill the pathogen or inhibit their virulence? And so we did a bunch of in vitro antagonism tests, and we couldn't find any evidence of direct killing. This is also a quorum quenching assay because our pathogen uses a quorum-dependent virulence mechanism, and the commensal can't quench the quorum signal of the pathogen. And so that starts to make us think that maybe it's something about the plant. So either the commensal is just more fit and growing in plant root exudates, or the plants can actually detect the pathogen and kill it, and not the commensal. And so this is recent work that we've just posted a preprint of from David Toms, who's a postdoc in the lab that's just started his own lab at Florida State. And so he sort of asked if the commensal and pathogen could if there were differences in kinetics in vitro versus in vivo. So again, asking, is it plant specific? He found that when he competed the pathogen and commensal in vitro, so our commensal is in gray, the pathogen is in blue. So in vitro, they always grow at the same rate. And so this is just total bacterial growth in monoculture. And then pathogen and commensal have the same final OD and very similar growth kinetics when competing in culture. However, in planta, our commensal always wins. And so here's the commensal in gray and the pathogen in blue. And so this suggests that the competition is indeed plant dependent. And again, can either be plant immune related or just that the commensal is better able to colonize plant roots. And so David wondered, first decided to test, could the plant distinguish these? And he used immunity reporters. So these are immune inducible genes, and the reporter lines developed by Nico Gildner's lab. And so they're nuclear localized transcriptional reporters that are turned on in response to MAMPs and DAMPs primarily. So David asked if the commensal and pathogen could activate these reporters and found that only the commensal could activate, or sorry, only the pathogen could activate the reporters. So that's here with these blue dots. So this indeed suggests that the plant can detect or distinguish the pathogen from the commensal. So the next question is, how could that happen? So both strains have similar MAMPs. They're 99% identical by 16S. They actually have the same flagellin sequence. All of their peptidoglycan biosynthesis genes look, indistinguishable as far as we can tell. So it seems unlikely they could tell them apart by MAMPs. Also, our pathogen doesn't even have a type 2 secretion system, so the effectors are not likely candidates. And so we're really interested in how the plant is able to sense the pathogen and mount this immune response. And so, as I mentioned, if we sort of return to our classic paradigm of immunity in plants, you have type 3 secretion where plants could recognize the effectors or the consequences of those effectors, but our pathogen doesn't have effectors. They also can recognize MAMPs and mounts immune response, but our MAMPs are probably very similar. And so it seems that that's unlikely. I think damage is probably our best bet because these are pore-forming toxins, and so they can cause damage to the plant cell, and then maybe the plant could recognize that. So to start to get at the answer to this, David started looking at what genes were responsible for this immune response. And so now I'm showing you, instead of a transcriptional reporter, a reactive oxygen burst, so another readout for immunity, where we have a dye that when oxidized, so the presence of reactive oxygen is luminescent. And so you get a peak when you get a reactive oxygen burst and an immune response. And so if you add flagellin, you get a strong reactive oxygen burst. If you add our pathogen and commensal, you actually get a weaker but still prominent reactive oxygen burst. And then this is just our baseline mock treatment. And so David asked, are there any plant genes in sensing of MAMPs or damage that are responsible for this reactive oxygen burst? And so the idea that if this is a MAMP response, like a response to flagellin, if you knock out components of PTI, and so we used a BAC1, BKK1, SERC1 triple mutant, which is a number of co-receptors for MAMP and DAMP signaling, we can see if this response goes away. And so first I'm showing you the commensal with the wild type or the triple BAC1, BKK1, SERC1 mutant. And so if you add flagellin, you get this peak, but then in the BAC1, BKK1, SERC1 mutant, it's down here at the bottom, the response completely goes away. And then our commensal, you get this peak in blue, and the response, again, like flagellin, completely went away in the triple mutants, suggesting the reactive oxygen burst we're seeing to the commensal is likely due to the presence of things like flagellin or peptidoglycan in our inoculation preps. However, when David did this with the pathogen, we saw a decrease, but not a complete loss of immune response. And so again, flagellin in gray, the commensal is in red, and we see a decrease in the response in the BAC1, in the BBC triple mutant, but it doesn't go down to baseline. So that suggests a little bit of the response is likely because of MAMPs, and then a secondary part of the response is independent of MAMPs, And I wanted to highlight that the back one is a co-receptor for a number of the damage receptors, but not all of them. So it does sort of rule out a subset of damage responses, but not damage completely. And so this suggests that pathogens or plants can distinguish pathogens from commensals. They can sense the commensals through MAMPs, and they can sense the pathogen through a second MAMP independent process. So we can see that the presence or the response is toxin dependent. And so this is, again, looking at gene expression now through qPCR instead of a reporter. And if you treat plants with, these are two separate reporters, so MIB51 and PR5, which are both immunity PTI responsive genes. Our pathogen induces a response. You can see PEP1, which is a damage associated molecular pattern, also induces a strong response. And these responses are decreased in the syringomycin syringopeptin mutant. And so this suggests that the plants are detecting the toxin or the consequences of the toxin, the damage from the toxin, to mount an immune response. And so we think that for commensals in the rhizosphere, plants can distinguish pathogens, but they may not rely on PTI or ETI to do so because we haven't found any role for those in responding to our pathogen. So it seems like they're responding to the toxins or potentially the damage those toxins cause to mount an immune response. And so to sort of summarize what I've told you, I've shown you that small genomic changes over very short evolutionary distances can result in really dramatic lifestyle differences in bacteria, and that plants have mechanisms to kind of sense their environments and then enrich for beneficial microbes. But then we think that when you're enriching for beneficial microbes, you need to have a way to avoid also enriching for pathogens. And so to do that, plants can potentially sense toxins or damage-causing molecules, and then hopefully, we're thinking that they can mount an immune response specifically to target those. I think a really open question there is, can they actually target their immune response to just the cells where those pathogens would be and causing damage? And so we are starting to think that plants can regulate the microbiome by sensing damage or toxins rather than MAMPs. And I think, this has some overlaps with the paradigms of immunity that I learned in my plant pathology class many years ago. And certainly damage and damage responses have some overlap with PTI. But I think we really haven't considered, what immunity looks like in roots. And given that all of these members of the microbiome have similar MAMPs, they may not have effectors. I think we need to start thinking about maybe some new paradigms. So how is it that plants could actually sense and regulate commensals to result in a community that's beneficial? Okay, so with that, I'd like to thank the folks in the lab that did the work. So I tried to highlight people as I went. The first story is the work of Ryan Melnick, a former postdoc. The FERONIA story is the work of Yi Song, another former postdoc. And then Nicole Wang and David Toms worked on sort of the last story on how plants can distinguish pathogens from commensals, and of course our funding, and I would be very happy to answer any questions.
Yeah, okay.
So if you find a way to transfer the two classes from commensals into the pathogen, will you predict the pathogen will lose its virulence?
Yeah, I really want to do those experiments. These islands are huge, so they've been hard to move around. But yeah, I think we would predict just based on the genomic sequence that the islands are incompatible in the same genome. And so if you tried to put them all together, they'd be lost. What could be driving that? I don't know. It may just be functional redundancy. So if both the syringomycin syringopeptin and DAPG are antifungal, then maybe you just don't need both, and so they're lost over time. But I don't think that's a strong enough selection pressure to explain the really dramatic pattern we see. And amazingly, we've started looking for this pattern now, and we see it with many other gene clusters. And so I think the most prominent example is with polyamine biosynthesis. They're just small molecules that are involved in signaling in bacteria. There's two pathways for spermidine biosynthesis that have been described, and Pseudomonas, all of them have one not both. And we see gain and loss over short distances. So I think we want to do that experiment with those islands because they're smaller and easier to move around. And then we can also select for them with ability to break down polyamines. But yeah, we've gotten really interested in like what could be driving these patterns because it seems too striking to just be kind of a, you get one and then you kind of lose the other over time. It seems like there needs to be active counter selection for one of them. Thanks. Yeah.
So I'm really curious. You brought up the suppressive soil phenomenon where a damage signal or a secondary, some kind of pathogen can allow for enrichment of.
Sorry, go ahead.
Sorry, so I'm curious if you've ever seen a case where a commensal or mutualist kind of mimics this pathogen signal, especially given the vast genomic data sets of some of the Pseudomonas species. If you can see, like, say, a RALF mimicking protein or something is being expressed by a commensal, because it seems like it'd be a huge fitness advantage and kind of like a cheating-esque fashion.
Yeah, so I guess you're asking in maybe in Pseudomonas or in commensal fungi, if they make RALF-like peptides. Yeah, I haven't looked, but that's a great question because it seems like commensal fungi would also benefit from, suppressing the JA pathway maybe a little bit. Yeah, that's a good question. I haven't looked. Yeah, sure. Sorry.
So do you think it's horizontal transfer for these islands?
Yeah, so we have been able, so the question is the gain and loss of the islands, is it through horizontal gene transfer? And it is, and it seems like it's horizontal gene transfer followed by homologous recombination. And so we can see a few instances, this is in the paper, that the island is more similar to the island in a distantly related species than, than the rest of the genome, I guess, or more closely related to distantly related species than to closely related strains. And we can even find examples where the island has been lost, leaving behind a lesion of homologous recombination, and the inside of the genes, right, where it's recombined, are more similar to a distantly related strain, and then the other gene is more closely related to closely related strains. So we can even see, like, it's happened recently enough that we can trace the inheritance and make gene trees of the genes in the island and the neighboring genes and see that they're moving around. Yeah.
So you think it's like some sort of integrative conjugative element? Is that like a plasmid, or is it extracellular and it's transforming into the bacteria?
We think it's just homologous recombination. So there's no evidence of transposon or like phage or anything else. Whether it's, conjugation followed by homologous recombination or transformation, we think it's probably conjugation because there's no evidence of phage. And then Pseudomonas are just not very naturally competent, but they conjugate really readily. So, but we can't tell how DNA is getting in, but once it's there, it's homologous recombination.
Sorry, one last, but have you sort of trawled through other metagenomic databases looking for similar islands that might have transferred to more distant?
Yeah, that's a good question. So I think David, a postdoc that's just started his own lab, is really interested in that. So toxins in general are so common in bacteria, and some of them are probably for intermicrobial warfare, and some of them are probably for hosts. And there are some examples, like within bacillus, of toxins moving around over short distances just when we look in the genome. But yeah, I think there's enough of these toxins that it could explain some of the differences in host association between closely related.
This specific one is pretty limited.
This one is limited to Pseudomonas. It's a pore-forming toxin, so there's many other pore-forming toxins that work through a similar mechanism, but are not similar. Yeah.
Thank you so much.
So I'm going to ask one question that came from Geneva. It says, thank you for an excellent talk. I'm curious how you captured these beautiful images of the Arabidopsis rhizosphere.
So I think maybe, do you mean you probably can't answer, but maybe these images from David on the immune reporters? If so, that we just have it's a fluorescence stereoscope that is actually not that expensive, that David was able to image these reporters on. So yeah, David's a cell biologist by training, so that's a lot of that is him.
Great. Well, thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.